I was going to go through the evolution chapter first, and if we get through it, then go into the case study paper. But I'd like to try and get through both of them today so that we can move into metagenomics for the rest of the class. So this will hopefully be the last class focusing just on genome sequencing. And what I basically wanted to do, and I have a few examples that aren't in the book chapter, is just take you through examples, some recent, some old, of how people are using evolutionary analysis in some way integrated with genome analyses. That's what sort of the last part of that chapter was about, studies of lateral gene transfer or whole genome phylogeny, and I have a few other ones to talk about. And I just thought I'd go through this really simple one because this is becoming a really important or common approach now that sequencing genomes is a lot cheaper and easier than it used to be. And the basic thing that people try to do is they take a data set for which they know something about the evolutionary history of the organism. So you start with a model about the evolutionary history of the organisms. And then you generate genome data such that you think you kind of basically bracket a major evolutionary event. Say, this is really common in studies of vertebrate evolution. For example, you get a bird genome and a mammal genome, or you get a marsupial genome and a placental mammal genome, and you try and basically bracket certain evolutionary events, and then you use comparisons of the genomes to each other and to other genomes to try and identify what happened at or around this major evolutionary transition. I'm just going to show you an old example of doing this, which was really sort of a classic example and a really useful result in studying genomes. And this happened sort of early in sequencing eukaryotic genomes. A group sequenced the second yeast genome, Schizosaccharomyces pombe, sort of the other model yeast compared to Saccharomyces cerevisiae. And they did a really nice comparison, which is they basically said, let's take all the genomes of eukaryotes and compare them to each other. And then compare those also to all the prokaryotic genomes. And let's ask, are there any genes found only in eukaryotes and found in all eukaryotes that are absent from the bacterial and archaeal genomes? And what they're trying to do here is, again, this is really getting much more common as it becomes easier to sequence genomes, is they have a model in their head. At the time, people believed in the three-domain tree. I'm still not sure where I feel about this, but there's a model that they have in their head, which is that there was a long branch leading up to the common ancestor of all eukaryotes. And then if you take, at the time, they took the Arabidopsis genome, I've labeled it here in black, and the human genome, and the fly genome, and the worm genome, and 2 yeast genomes. So they took a bunch of eukaryotic genomes, compared them all to each other, and if they all share some genes with each other, those genes should have been present in the common ancestor of all those eukaryotes. That's the simplest model to explain, when they all have genes in common. And then, if those are genes that are absent from all archaea and all bacteria for which genomes are available, the simplest model is that those genes originated on the branch leading up to the common ancestor of all eukaryotes. And so that's what they were basically doing, is doing a simple genome comparison to find genes that originated on this branch. And lots of people are interested in this because these are like the hallmark genes of eukaryotes, the genes that originated on the branch leading up to the common ancestor of eukaryotes. Does that make sense, basically, what they're doing here? And so this is really cool. They found about 200 genes shared by I forget how many genomes, the eukaryotic genomes they looked at, something like 8. And they nailed it in terms of these are genes that you sort of expect. They're the hallmark genes of eukaryotes. So they found cytoskeleton components, one of the key features that eukaryotes have that bacteria and archaea don't. Tubulin and myosin and other cytoskeleton components. There are components of the chromatin and DNA packaging sort of systems, and a lot of other genes that are associated with sort of nuclear processes. And many of these genes had no known functions. This is a really great sort of, if you want to start a biotech company or something of that effect, you find, I forget how many it was, but 50 genes that are conserved across all eukaryotes, absent from all bacteria, and no one had done an experiment on any of them. Really good candidates for interesting conserved eukaryotic processes. So I thought this was really cool when they did this. And then they went the next step and they asked a similar question, but now comparing the different eukaryotes to each other. So they said, okay, let's do the same thing, but now let's compare the multicellular eukaryotes to the single-cell eukaryotes and try and identify genes that are responsible for multicellularity. And they found 3 genes conserved in the multicellular eukaryotes that they looked at the time, which was Arabidopsis, Drosophila human, C. elegans maybe. And they concluded from this that multicellularity was not due to the invention of new genes, but was more likely to be due to regulatory processes. This is consistent with everybody says about how eukaryotes diversify by novel regulatory processes. Now, it turns out this is completely flawed, this second analysis. It is completely useless. Does anybody know why? True, but let's pretend like that.
Are you getting around to just the presence or absence of homologs? Is that all?
That's what they did, just presence and absence of homologs. But they made a mistake here. So this analysis, where they were looking at across all eukaryotes to try and find things that happened on this branch. Now let's ask about multicellularity. So where on this tree would the genes have evolved that led to multicellularity if you have plants here, animals here, and fungi up here? What the problem is that multicellularity has evolved multiple times, separately in plants versus animals. And so, well, duh, you're not going to find genes that are shared among these groups that are responsible for multicellularity when it's convergent evolution of a process. So on the one hand, they did this brilliant analysis of finding genes responsible for sort of eukaryotic core processes. And then they just didn't think through the model for the second approach, which was find genes responsible for multicellularity. There's no way to find anything interesting from this sort of second analysis. Does that basically make sense? And this is, again, it's an old example, but this is what everybody is doing now when they're sequencing genomes across the diversity of life, asking, can I find things that are responsible for the origin of flowers and comparing angiosperms to other types of plants, and et cetera. And you have to always make sure that what you're looking for has a place on a tree, that you're trying to detect something that is sort of consistent with the evolutionary model. And if you, for example, people are comparing, and they're doing it correctly, they're comparing different groups of mammals that have evolved obligate herbivory, that they only eat plants. They're not looking for genes shared in common across these mammals because they're just 25 different cases across the mammalian tree. They're looking for other types of convergent evolution, like the number of copies of certain types of genes that you might have, not the presence of a particular gene. Yeah,
did you think hemoglobin and leg hemoglobin are completely separate radiations, or completely separate
yeah, so I don't know. I mean, plants have a protein called leg hemoglobin that many people think is homologous to the globins of animals. I don't know enough about it to know where I would, But
it'd be possible to argue either way on that.
So if you have genes that are similar to each other across distantly related taxa. There are basically 3 models to explain that. They were present in a common ancestor and lost from organisms that don't have those genes. They were absent from a common ancestor, and one of them acquired them by lateral gene transfer, which we'll get to an example of exactly that at the end of class, but not with hemoglobins. Or a third possibility is that they're not actually homologous, and they have become similar in sequence by convergent evolution due to constraints like the need to bind oxygen in water-dissolved solution, which globins are really good at.
But you're going to get to that later. So, right?
I won't talk about hemoglobin, but I'll give an example of just an absurd example of lateral gene transfer to explain one of these cases that it seemed so ridiculous when people proposed it, but it turns out to be lateral gene transfer. So I just thought I'd go through a second example because there are a few in the book chapter. It's not really that endosymbionts per se are interesting. I mean, I'm interested in them, but they're a good example of how you can use whole genome evolutionary analyses to ask questions about the biology of organisms. And so endosymbionts are organisms that live inside the cells of another organism. Usually, I mean, most of the ones that people have studied extensively are bacteria that live inside the cells of eukaryotes, but there are eukaryotes that live inside the cells of eukaryotes, and there are a couple of examples of bacteria that live inside the cells of other bacteria. And when people have studied, in particular, the bacterial endosymbionts, they found if you compare those endosymbionts to free-living organisms, so close relatives that don't live inside the cells of another organism, the endosymbionts have a smaller genome than their free-living relative. They have a lower fraction of G's and C's in the DNA, lower GC content than their free-living relatives. PI is the proportion, it's a measure of the acidity, basically, of protein content. They have a higher PI, and they have higher rates of evolution. And all these things together, and this is just one symbiont that I'll show you examples of, but this is true for every endosymbiont that anybody has really looked at. They have these 4 features. And what people have done a lot of as they've gotten genome data is to start to ask, can we use sort of deeper genome analyses to tease apart what's going on? Why would endosymbionts have these 4 features? And this is just an example. So one thing you can do to test this one part of it that says they have higher rates of sequence evolution, this was originally developed by people looking at like one gene. And you build an evolutionary tree of that one gene, and the branch leading up to the endosymbiont is really long, and all of the free-living relatives, the branches are shorter. So what this means when you have a long branch is that since the endosymbiont shared a common ancestor with this free-living organism, at some point the rate of evolution went crazy in the endosymbiont. And when you get whole genome data, you can show that this is the case across whole genomes. So this is from a study by Nancy Moran's lab where they did this. This is the gamma proteobacteria. These are 2 endosymbionts, very close relatives to E. coli, but their evolutionary rate has gone kind of crazy. This is just another example of that. And so when people were studying this, they basically came up with 2 hypotheses to explain this. One is that, so this includes all of the features of endosymbiont evolution. So you have a free-living bacteria, and then at some point one of them moves into become an endosymbiont, and then all these weird things happen after it becomes an endosymbiont. It loses genes, that's why its genome gets smaller. Its GC content goes down, its evolutionary rate goes up. So why does this happen? And there are these 2 models, basically, that people came up with. One is that when these endosymbionts lose genes, it may just be sort of random. Endosymbionts don't get exposed to a large population to engage in recombination with. They may just not be able to recover genes once they lose them that free-living organisms do. You throw away some genes, and one of the types of genes that you throw away are genes that are involved in replicating or repairing your genome whenever it gets sort of damaged. And if you lose, among the genes that you lose, if you lose repair and replication genes, eventually that may screw up your GC content, because these are the things that control your DNA, and it may also lead to your evolutionary rate to go up really high. And that in turn probably leads to the organisms to become really weird, and lots of weird things happen. And the alternative model is basically what's called the population genetics model, which is that endosymbionts, when they get passed down over, if this is in an amoeba or in an animal, when they get passed down to the next generation, they usually go through a really small population bottleneck with a very small number of individuals that lead to the next generation. This basically leads to genetic drift, the equivalent of what happens with inbreeding or other types of sort of weird evolutionary events. Genetic drift basically causes organisms for selection to be inefficient. And what this will do is you might, if there's a mutation that normally would get weeded out by selection, it might survive, because what genetic drift basically means randomness becomes more important. So mutations stay around in the population more, the rate of evolution goes up. Mutations stay around longer, you may eventually knock out one of your DNA repair genes, and the GC content might get screwed up, and so on. So there are these sort of two related but conflicting hypotheses. And most people that were working on endosymbionts argued that the population genetic explanation was favored. And this was in part by doing genome sequencing and finding that many endosymbionts actually with long branch lengths and low GC contents had, as far as people could tell, a full suite of DNA repair genes in their genome. And so people favored the population genetic explanation at least a little bit. But if you compare endosymbionts to each other, you see a really interesting thing. So for example, these are all 6 different endosymbionts. And if you build evolutionary trees of all of their genes in the genome, you see that these 2 in particular, this is an endosymbiont of the tsetse fly, causes sleeping sickness, and this is an endosymbiont, I think, of an ant. And they have longer branch lengths than these endosymbionts of aphids or this endosymbiont of this glassy winged sharpshooter. And if you look at now DNA repair genes in the genomes of these, you find that the ones with the lower evolutionary rates have the genes that are involved in correcting DNA replication errors, and the ones with the higher evolutionary rates are missing certain DNA repair genes. So what's probably going on is that the big difference between endosymbionts and free-living organisms is more related to drift and population bottlenecks. But among endosymbionts, the ones with the really high evolutionary rates tend to be the ones with really weird DNA repair processes. So it's both explanations are true. And by having genome data and integrating it with a phylogeny and information about the organisms and their free-living relatives, you can start to test models that would explain what's going on in these organisms. And this is, it's not trying to argue that endosymbionts are different than anything else. This is what people are now doing with genome data related to virulence, pathogenicity, photosynthesis, all sorts of processes. You're able to integrate information across phylogeny, presence and absence of genes, different features of the organisms, where they're found, et cetera, and whole-scale genome data. And basically test. You're not doing an experiment here, but you can test models that explain the biology and evolution of these organisms by trying to decouple the different features. So it turns out, I lied to you a little bit. So it turns out that these are not a perfectly correlated set of features. There are some endosymbionts that don't have lower GC content than their free-living relatives. It's very rare. But by looking for these cases where one of these things is decoupled from the rest, you can start to try and understand maybe that endosymbiont, for example, doesn't go through as many population bottlenecks as the other ones. Or there's actually, all of these features are found in organisms that are not endosymbionts, but are maternally transmitted from mother to offspring. So there are these beetle symbionts that are not inside cells, but they're between cells, and they have all of these features. So by starting to decouple, all these bits of information, you can actually retrospectively test hypotheses about how they got created and the mechanisms of sort of replication, repair, endosymbiosis, et cetera, in these organisms. That basically, the endosymbionts are just an example of this. And again, as people get genome data, you're able to develop a test like this retrospectively for all sorts of interesting processes. Yeah?
So I guess this would be sort of hard to do because when you're looking at a tree, you're sort of looking at a snapshot in time. But is there a way to tell the sort of the rate of the rate? Like if you were following a population and you wanted to see, I guess, whether the rate of evolution was increasing faster when the endosymbiont was.
So if you fill in a tree, you can, for example, find cases like here. The rate of evolution for this whole clade seems to have gone up sometime in their ancestry. And then there was an increase and an increase again. So yeah, you can basically, by taking modern data, you can infer...
So sort of previous branch lengths.
Not just previous branch lengths, but you can infer actually what the genome looked like in the ancestor. What genes were present in the ancestor. You can infer...
That would just be a consensus sequence or something?
No, what most people do is they treat genes as traits. And then you use character state reconstruction methods, just as you would try and infer what the bone structure was of an ancestral vertebrate. You can just have gene 1, present, 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 absent, 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 absent. And that, the simplest model is that it was present in the common ancestor of those three. You use parsimony, character state reconstruction, or if you're tricky, you use a maximum likelihood method. But you can test, and people actually reconstruct whole genomes of organisms in the past. They're a model, but as you get better and better sampling, your model for what the ancestral genome looked like gets better and better. And then you can test basically what you're saying. You can integrate across the whole tree over time and test your model not just for the tips, but for the entire tree, for any type of data, whether it's evolutionary rates or photosynthesis. This is how people try to figure out what was going on in terms of the rate of oxygen production by cyanobacteria at the time of the great oxygenation of the planet by doing this type of thing. Any other? So a third sort of very common issue in integrating evolution and genome analysis is this issue of lateral gene transfer, which we've sort of briefly talked about, but not talked about in a lot of detail. So the sort of main thread of inheritance of organisms has always been this model of vertical evolution. That is, you transmit traits from parent to offspring, and then you extend that concept to species transmitting traits to descendant species, and eventually phyla transmitting traits to all the members of the phyla across, an entire tree. And you draw the tree of life as this vertical where the inheritance of traits is from the bottom to the top, and that's why it's called vertical evolution, or vertical inheritance. And, this makes sense, right? Organisms replicate their genomes, they transmit their traits to the offspring cells, even in sexual reproduction, it's, you know, complicated because there's merging within a population, but parents transmit their traits to their offspring. This is pretty simple. And it turns out that this is how people thought everything worked until, you know, the 1940s or so, and people started to do experiments primarily in bacteria and discovered that vertical inheritance is not really the only thing that's going on. And thus there's been this sort of concept, it's a lot of different things going on at once, of what's called horizontal DNA moving from one branch to another. Opposite of vertical evolution has been called horizontal gene transfer, or lateral, sort of the same thing, lateral inheritance. And there are a lot of different examples of this where we know the mechanism by which it can occur in different organisms. So one of them is this competence. Unclear how common this is across lots of organisms, but basically it's a process by which an organism just takes up DNA from the environment and the DNA gets integrated into its genome. There are some bacterial species that do this sort of routinely as a part of their life cycle. And just generally called competence. There's conjugation in a lot of bacteria. This is in some ways the equivalent of sexual reproduction, although it doesn't work in exactly the same way. Basically, two cells will form a little bridge between them. DNA gets passed through the bridge. Sometimes this DNA is duplicated, so a copy stays in the donor and the recipient. Sometimes the DNA actually just moves from one cell to the other and doesn't stay in the donor. But it's basically a mechanism by which bacteria can exchange, bacteria and archaea, and even some eukaryotic cells can exchange DNA from one cell line to another. Conjugation can move DNA across large evolutionary distances. This example came up, I think you asked about this before. Agrobacterium does this to plants. So it forms a little pilus, basically transfers its DNA into a plant host. The DNA gets integrated into the genome. That's moving DNA from a bacteria to a plant. And there are many other examples of DNA moving by sort of a similar general concept of conjugation across really big evolutionary distances.
Is there any anecdotal instances of that happening, either agrobacterium or anything else, into the human genome that potentially occurred?
There are no good examples of this happening in humans. There are plenty of good examples of the next process happening in humans, which is viral transduction. So transduction is usually sort of reserved, I'm not even sure, maybe Holly knows, for cases where it's a bacteria-based or archaeal-based process. But basically, the mechanism is as follows. You have a cell that gets infected with a virus. Viruses take over the machinery of the cell. They're not able to carry out all cellular processes themselves. They're parasites. They take over the machinery of a cell. And one of the things that they eventually do is they get the cell to make the outer coat of the virus, the capsid, and they get the cell to make copies of the viral genome, and then the virus is supposed to package these together, put some of its genome into one of these capsids, and then pop the cell, and the virus then can spread to other cells. Occasionally, the virus will put into its capsid DNA that did not come from the virus. Sometimes it's a part virus DNA and part bacterial host DNA. Sometimes it's just all host DNA. And now that's a virus that functions. It's got all the proteins and all the machinery to float around and attack another cell, but its DNA is actually DNA from the host rather than a normal viral DNA. And now it can go infect another cell and inject its DNA into that other cell, and now you basically serves as a vector to transfer DNA from one cell to another cell rather than moving viral DNA. And this can move DNA across small or large distances, evolutionary distances, depending on the specificity of the virus, what types of cells it's able to infect and then transfer the DNA to. But there are many examples of this type of thing happening in plants and animals, where viruses inject their DNA into plant and animal hosts, and the same type of thing can happen where they can occasionally move the wrong DNA around. I think it's a lot less common, in part because for a virus to, well, it's a lot less common in multicellular organisms, because for a virus to lead to sort of long-term transfer of DNA into a new host lineage, it has to get the DNA into some type of germ cell line, right? It doesn't, if it infects your skin, even if you get some, like an agrobacterium, some tumor in the root, it has to get into, you have to convert that material into some germ lineage in order for it to get transferred to the next generation. In plants, it's pretty common. Most plant cells are reasonably totipotent, and they can lead to a whole other plant. In humans, if I got an infection by some weird virus and it led to a tumor, those aren't going to turn into germ cells most of the time and lead to reproduction. So it's not as common between generations, but it is a major mechanism of tumor formation in animals. Transfer of DNA and movement of DNA from viruses, and even occasionally from one host to another host. Any other questions about that, or does that basically answer the...
Just curious.
So there's a new paper, which we're not going to go into, that scanned through human genome data and claims that they found examples. Again, these are all somatic. This is not saying these are herited from parent to offspring, but in genome data that was generated mostly from cancers in human populations or human individuals, they found a reasonable number of examples of bacterial DNA integrated into the genome of these cancer cells. And they suggested that maybe bacterial conjugation with human cells was more common than it was appreciated. But that's the only example. It just recently came out, and it's the only example I know of that.
Other than the any school or the Margellons people.
I don't even know what that is.
Oh boy, it's real fringy.
And so if we go back, I'm not going to show the slides, but if we go back to that discussion of E. coli, O157, H7 versus K12 versus the other E. colis, and the lots of variation that you see among closely related bacterial or archaeal species, in many cases we see these islands of DNA in one strain and not in the other strain. Very frequently, those islands contain pieces of DNA that correspond to viral components. So they may actually represent viral genomes that have inserted into the host genome, or portions of viral genomes that have inserted into, for example, the E. coli O157H7 genome. You can't always tell what the mechanism was leading to weird DNA in one organism or another, but certainly viruses play an important role in this. Anybody know what time it is? Okay. So lateral gene transfer, it's unclear how big of a deal this is across all of evolutionary time. So if you ask the question, if I integrate across a tree, and I take a group of organisms and I say, what is the rate of vertical evolution versus lateral or horizontal evolution in that group? It's most of the studies that have been done, and I'll give you a figure from the chapter has an example of a good study, but most of the studies that have been done haven't really tried to figure this out. And so it's been unclear in most groups of organisms what the balance is between vertical evolution and this lateral gene transfer. We certainly know many examples of lateral gene transfer being important in certain situations. Antibiotic resistance is the best example of this. Plasmids make small pieces of DNA can make an organism resistant to certain antibiotics. Those small pieces of DNA can get exchanged to other lineages by conjugation or competence mechanisms. So it clearly happens, but the extent to which it happens is not entirely clear. And I don't want to spend a lot of time on this, but basically the way to figure out the extent of what's going on in any organism is to start to develop models that sort of model vertical evolution versus horizontal evolution, what you would expect them to do to the DNA in an organism's genome, and then take genome data and try and deconvolute from that data. You know, if you saw something like this, where these two organisms had that gene, this one didn't, this one didn't, this one didn't, and this one didn't, is that it was present in the common ancestor and lost from a few lineages, or is it that it originated over here and got transferred over there? And if you have a good model for what you expect to see if one occurred versus the other, you can tease them apart. And I'm just going to, oh, sorry. So basically what most people, when they've thought about these models, they think about things like that two different lineages of organisms, the DNA, even if they keep the same genes in their genomes, the content of those genes starts to drift from one organism to another. So the GC content of an organism's genome becomes different, maybe by completely random forces. The codon preference in protein coding genes becomes different. And a few other things that you could call sort of genome flavors or genome features become different. And that's important because after a transfer occurs, and if that transfer stays around, if it gets integrated into the genome of a new recipient and then transmitted to the offspring, it should, at least for some period of time, look in many ways more like the donor in terms of these flavors than it does like the recipient. And it should also look phylogenetically incongruent with the phylogeny of the rest of the genes in the genome. And so you can start to build up sort of models for what you would expect to see. And if you apply one of these models, that's what someone did with this analysis, they calculated not over time, but a snapshot for each individual genome, what fraction of the genes appeared to be laterally transferred versus vertically transferred. And there are big differences between organisms, not that surprising. There's, if I go back to this, after genes get transferred, eventually they're going to start to acquire the genome flavors of their new host. So this is called amelioration, and they lose that distinction. So if they had a high GC content when they got transferred from a high GC organism to a low GC organism, eventually they're going to become low GC content, even if they stay around. And people have estimated how long that takes, but you can use that to try and tease apart if you find a gene that seems anomalous. If it was a recent lateral gene transfer, it probably should have a weird GC content or composition.
You can't really use that if it happened further back.
Right, I thought I had a slide about this, but apparently I ditched it. Right, so the problem you have with doing a lot of these analyses is that the timing of when you can, when the event occurred and whether or not you can infer it with a particular method varies. So if a gene was transferred from cyanobacteria into a plant, even if it lost its GC content bias that you saw in the cyanobacteria over, let's say, 20 million years, phylogenetically, it's going to look like a bacterial gene. Whenever you build an evolutionary tree, it should stick in an evolutionary tree where bacterial genes normally stick. But phylogenetic trees are a terrible way to detect very recent lateral gene transfer events. So each method has its own biases and flavors, and to really tease all of this apart, you need a model that allows for all of them to scan through the data. So you're going to use GC content to look at recent lateral gene transfers and phylogenetic trees to look at old lateral gene transfers and other things to look at medium. Does that make sense? It's medium scale. And that's basically what was done here. So this paper also from Nancy Moran's lab, where they took 12 or so genomes of gamma proteobacteria. They compared them all to each other, identified all the genes that they shared in common or that they didn't share in common. They built an evolutionary tree of the core, the 200 genes that all these genomes shared in common. They built a tree. of those 200 genes that they all shared in common, so these are the core genes across all of them, they all showed the same phylogenetic tree, basically. So those core genes reflected sort of the central vertical evolution of these genomes. And then many genes didn't reflect that. And so they drew this really nice figure where if you have little lines going in, those are additions of genes into one lineage. And they also tried to draw loss of genes in one lineage. So they basically took presence and absence of genes, a model for how to figure out whether or not they were present in the ancestor or laterally transferred into a lineage, and came up with a complete model to explain the presence and absence of all the genes in each genome over time. I don't think they used GC content here, but they could add that on to their analysis. It's a lot easier to detect lateral gene transfers when they've happened across, yeah.
Oh, it just seems like there's always going to be a resolution problem, right? Like the closer phylogenetically you are to the thing that horizontally transferred to you, it's like there's a limit to the resolution.
So fortunately, it's less interesting to find those cases, right? So if there was a lateral gene transfer between close relatives, that is more similar to eukaryotic sexual reproduction than it is something weird. So it's still useful to infer those cases but that's basically more like recombination within a population, even if the population might be genera level instead of species level. So, but there are new papers where people have models that try to tease apart recombination within a species as well as lateral gene transfer between distant relatives. And it is true that it's different methods that detect recombination within species than are used to detect lateral gene transfer. I just showed this here because it's really easy to detect lateral gene transfers from organelles into the nucleus of eukaryotes, mostly because they're so different than the normal nuclear genes that it's just a lot easier to detect. So here's so there's the suite of methods that you could use to detect gene transfers, including where you find the gene, what the nucleotide composition is, just whether or not its sequence is really similar to something that is supposedly distantly related, unusual phylogenetic trees, and the last, which people are doing more and more of, is actually observe, really in a population, the gene transfer events themselves. Okay, so here's the case study I want to show you, which just still blows my mind. It's completely ridiculous. So it's this case study of aphids. So there are these pea aphids, and they have this color polymorphism, basically, in some of the aphids that people have studied, where some of them are green and then some of them have this orange appearance. And if you do basically a biochemical characterization of the lipids and other components from these organisms, you see that the ones that have different colors have different relative abundance of certain carotenoids, which it's kind of weird because animals don't make carotenoids. That's why we have to eat, things that have beta carotene in them, because we biochemically know animals are known to make their own carotenoids. And so people sort of saw this, they didn't really pay that much attention to it, but then the genome of one of these pea aphids was sequenced. And Nancy Moran, again, I didn't purposefully pick papers only from Nancy Moran, but she is absurdly brilliant. The genome was sequenced of this aphid, and Nancy Moran was asked to help look at the genome because she works on the endosymbionts that live inside aphids. So aphids are obligate sap-feeding insects. Sap from the plants that they feed on usually has the essential amino acids removed from it so that animals that only eat the sap can't survive. Unfortunately for the plants, aphids have evolved this symbiosis with bacteria that make all the missing amino acids that they don't get from their diet, and Nancy works on those symbionts. And then the people who were working on the aphid said, we sequenced the genome, here's the list of genes, if you find anything interesting related to symbioses, take a look at this, and at least I think that's how it played out, basically. And she saw on this list these genes annotated, you did blast searches and someone annotated them as carotenoid synthase. And most of the people didn't really realize that that's just not supposed to be in an animal genome. Long story short, they went, looked at the genome data. It looked like these are really in the genome. That's what this annotation says down here. It wasn't like a contaminant from the genome sequencing project. People did genetics where they showed that it was transmitted from, aphid to offspring. If you treat them with antibiotics, I think it's still transmitted from aphid to offspring, so it didn't appear to be in some weird bacteria. They then eventually, characterized these in more detail, built evolutionary trees of them, and here are the aphid sequences from different aphids, and they're nestled in an evolutionary tree among carotenoid synthesis genes from fungi. And what happened here, although they don't have a good mechanism for this, is that somehow, maybe because the aphids used to have a fungal symbiont, even though now they mostly rely on bacterial symbionts, they stole the genes for making carotenoids from a fungus. Those somehow got integrated into the genome and somehow got transmitted to offspring such that it now becomes a heritable trait. Before this, I didn't know of any good examples of lateral gene transfer from a microbe into an animal that got transmitted from generation to generation. There are now many more that have been found. So it's much more common. Maybe the fungi was infecting germ cells. I mean, it's not clear what was going on here. But these aphids are able to make their own carotenoids with genes that they've stolen from carotenoid synthesizing fungi. It's pretty weird. If people are interested, there are a couple of new papers from that same group where they've characterized this in more detail. And every time it's just more and more bizarre and fascinating. So are there any in the humans? There are still no good papers that I know of saying that there are human genes that came from microbes sometime, relatively recently during evolution, but this certainly suggests that it's more possible than I would have thought beforehand. Okay, so I want to switch to, anybody give the time?
3.
It's 3?
It's
To
4.
4. I want to switch to talking about this genome encyclopedia study as sort of a model for evolutionary, another model of evolutionary genomics. Anybody have any other questions about lateral gene transfer or weird aphids? Okay, so this is a pretty simple concept that has been used in many genomic studies, although because of this issue of lateral gene transfer and the reason I wanted to do them sort of closely together, people have not done this or didn't think that this would be worth doing in microbes as much as in other organisms. So the concept is you look at a tree of organisms, whatever level you want to look at, and you say, if I'm going to sequence genomes of these organisms, it would be useful to sequence organisms from across the diversity of this phylogenetic group. And this was first done in sort of big scale with the NHGRI, the National Human Genome Research Institute, said, we're not just going to focus on sequencing the human genome, but to understand the human genome, we need to sequence and analyze many other animal genomes to compare to the human genome. And they basically set up sort of a tiered approach where they said, we want to do some primates. Those will give us the close relatives to compare to identify features that we share with our close relatives, and that'll allow also the identification of what's new in humans compared to primates, other primates. And let's also do a collection of other placental mammals so that we can sample different levels of distance from the human genome. And then we should do some birds, we should do some reptiles, we should do some fish, we should do some insects, and so on. And they focused on model organisms for many of these, but they also said we need to sample the tree of vertebrates and the tree of animals in order to best understand the human genome. And for plant studies, this has also been done where people said, we're not just going to characterize the model plants, but in order to understand the maize genome, we have to have a bunch of other grass genomes, and in order to understand the pine genome, we have to have a bunch of other conifer genomes, and so on. It's sort of a very straightforward concept to say phylogenetic diversity is an important component of genome sequencing. And no one was really doing this for microbes. And I think the main reason, there are two main reasons for this. The diversity is very large, so people freaked out. It was expensive, and there's so many friggin' microbes. Why would you try and cover their diversity? And the second issue was this issue of lateral gene transfer. So if lateral gene transfer occurs at a very high rate, then what does a tree mean, right? I mean, so we haven't really talked about this, but if you start to add lots and lots of horizontal movement of DNA, this is not a tree anymore. And it clearly isn't. So we call it a tree of life. It is a web of life. There is no single, each organism, modern organism, different parts of its genome probably have hundreds of different histories. That we have lots of organelle-derived genes in our genome. We've undergone, probably a few lateral gene transfer events relatively recently. There certainly were many a long time ago. Plants have all these chloroplast genes. There's lots of stuff going on. And people went the next step, I think, which said, okay, there's so much that there's no vertical evolution. That is, that there's no, if you went and sampled something that's called cyanobacteria, because its ribosomal RNA shows it grouping with other organisms that we call cyanobacteria. If we looked at any other gene in the genome, it's going to show a different thing. It's going to say that cyanobacteria 11 is actually, if we looked at ribosomal protein L4, cyanobacteria 11 is actually closely related to spirochetes. And people had this, I think, this feeling that this would swamp out the signal of vertical evolution. Turns out that's not the case. So if you go back to this Moran study, which was one of the first big-scale studies to show this, 200 genes shared across these genomes, they all have the exact same tree, basically. Lateral transfer is not swamping out the signal of vertical evolution. It is just on top. There are some lateral gene transfer events, but they don't totally swamp out your signal. And what this means is that basically, if you want to discover diversity, if you want to characterize sort of all the diversity of different protein families in life, a potentially good way to do this would be to say, okay, we've sequenced 10 cyanobacterial genomes. We don't need that many more of those. There are 100 other phyla we've never sampled. Why don't we sequence some of those? And we made this argument many years ago when I was at my previous job. So this is the model of what genomes were available at the time. There were 8 phyla of bacteria that had no genome sequences available, but had cultured organisms, cultured representatives. So we said these should be sequenced, and we got a grant to do this. And at the time, that was basically half or so of represented about half of the total diversity of bacterial genomes that would be available. And then at the end of this project, it was clear that genomes were still really poorly sampled. And I moved to here, and I convinced the Joint Genome Institute, which is in Walnut Creek and is a Department of Energy genomics facility, to go through the tree of bacteria and archaea and identify branches that had no genome sequence available, but a cultured representative, and just start sequencing them. And they've done about 300 of these now, maybe more. It's hard to keep up. But the idea was that people were sequencing genomes based upon whether or not they killed someone. People were sequencing genomes based upon whether or not they were known to have an interesting biological property. People were sequencing genomes based upon whether or not they came from an interesting environment. The only thing people weren't doing for bacteria and archaea was sequencing phylogenetic diversity. And I mean, I don't know how, I could guess why the Department of Energy somehow got interested in this, but it's not really an energy-related project, but that's okay. So they agreed to do this. We collaborated with this culture collection because none of us knew how to grow any of these organisms, but there was this great culture collection with this guy, Hans-Peter Klenk, who turns out to be an evolutionary genomicist working on microbes at a culture collection. And he said, sure, we could grow all these organisms for you for free. And we could even isolate the DNA for you and characterize it and FedEx it to the Joint Genome Institute for no charge. We asked the American culture collection, there's a thing called ATCC, the American Type Culture Collection, if they would be willing to partner with us. And they said, sure, $4,000 an organism. And I had a budget of $0 to order DNA, so we chose the free source. And I just have a couple of slides on this, basically. What are the lessons that were learned from this first phase of this project, which we published a paper a few years ago on? And so the first relates to a very simple question, which is, what we did was we went through the ribosomal RNA tree. That's where the most data is. We picked branches that appeared to be novel, but contained a cultured representative that was at the German culture collection, and sequenced it. And then when we were done with the genome, we built a tree of all the available genomes based upon one of those concatenations, like we talked about before, of a set of phylogenetic marker genes. And we asked, in this tree of the genomes, do the organisms that we picked still appear to be unique relative to the organisms, relative to the ribosomal RNA? It could have been that organisms would have a novel ribosomal RNA and their genomes would be completely boring and closely related to something else. That was not the case. So novel ribosomal RNA means, on average, the core housekeeping genes in the genome are also going to be novel. I mean, if that didn't work, we should have scrapped the whole project. I mean, admittedly, we did this after we did the first phase of the project. But, so another thing to realize, which we have not talked about here, is that the 16S tree is not always good. It's not always an indicator of the core genome evolution of an organism. So for example, here's an example, Hyphomonas, a bacteria that was of interest because it had interesting developmental processes. If you build a tree based upon the 16S ribosomal RNA, which was the only thing that had been sequenced before the genome was sequenced, it got classified in this order, the Rhodobacterales. And that confused a lot of people because its developmental properties were really different than these other organisms, and it led to some serious confusion about sort of mechanisms and sort of in the model of that S-POMB thing that I talked about. If you build a tree based upon the whole genome, or the 23S ribosomal RNA, it goes in a totally different, well, not total, a different order within this phylum, along with an organism called Colobacter, which also develops in a similar way to this Hyphomonas. So by building a tree with the 23S ribosomal RNA or the genome, it goes to a different place. The 16S was misleading. And this is not totally rare. I mean, it's relatively rare. It's not that the 16S said that this was, really distantly related from everything, and the 23S said it's E. coli. It's basically the resolution in deep branches in the phylogenetic tree with 16S is not always accurate. Yeah.
On that one, so the branch looks like it has a 52 bootstrap support on the 16S tree, and then it's like 100 on the other, the 23S.
Yeah.
Is that something you guys saw commonly, like the bootstrap support would go way up when you looked at other?
So it is common for bootstrap support to be low for individual genes. Mm-hmm. And that's the bootstrap support for 30 genes.
Oh, okay.
So by adding much more data,
yeah.
It's probably an unfair comparison. What we should do is compare bootstrap support with the same number of positions in the alignment, and then I don't know, maybe 16S might, that's still pretty high for deep branches in a bacterial tree. And previously, everybody else who built 16S trees for this organism, even with thousands of other taxa, still put it into the Rhodobacters. There's something weird about its 16S, probably lateral gene transfer, even though it's not supposed to occur for some of these genes. There's something happened with this organism 16S. So bootstrap values can hint at problems, but you'd have to do a, a more detailed comparison than we did here. And this is what we did basically to make the Department of Energy happy. Basically, the argument that we made at the beginning was to copy the argument made for the human genome, which was that if you have humans, chimp and gorilla, and then march your way down through the tree of mammals and vertebrates, that by having different degrees of distance between the human genome and other genomes, it will help you annotate different features of the human genome. So if you want to find what's conserved across all mammals, you need a good sampling of mammal genomes. If you want to find what's conserved across all eukaryotes, you need a good sampling of eukaryotes, and so on. And we basically showed that the same is true for bacteria, that there are all sorts of aspects of annotating genomes that having phylogenetic diversity helps with. Again, if lateral gene transfer wiped out our signal of evolution, that would not have been the case. I'm just going to skip that, and then this is probably the last thing we'll talk about, which is the number one thing I think that came out of this project is really the counter to lateral gene transfer. And I'm going to... So if you sample organisms by where they sit on the ribosomal RNA tree, and you ask, what is the probability that they're going to have something new compared to what we've looked at before? If there is no lateral gene transfer, then the probability that they're going to have something new relates to things like invention, the probability that they invent a new function in their lineage versus convergent evolution. So you could have, an organism over here that lives in the same environment as an organism over here, and they could have converged upon many solutions like growing at high temperatures or growing at high pH or low pH or something to that effect. And if you wanted to find, ways that organisms live at high pH, then, sampling organisms that live at high pH, even if they're distantly related or closely related, could be a way of doing that. But if there's lots of lateral gene transfer, then the probability that you find something new is mostly going to relate to whether or not it's encountered lots of other organisms that have invented other things sometime in its history. And that's what we were told when this was reviewed, was that there's enough lateral gene transfer that you're not going to find anything new by this approach. The probability that you find something new in a phylogenetically novel organism is irrelevant. What's going to be important is where they live. What is their selective pressure for their environment? And whether or not they could have, and then if they live in a stressful selective environment, they would have had enough time to steal genes from wherever they could have gotten the right genes, such that their phylogeny isn't going to matter. And we did a really simple test of this, which is we basically asked, if you take a genome and then you take another genome and you ask, what is the probability that they have different gene families in those genomes? And you ask this if you take two genomes and then you take a third genome. And you do this for different sets of genomes. So you do it for a set of closely related genomes. So let's say you do just the close relatives of this one cyanobacteria, and you do 10 genomes, and you do a rarefaction curve for the number of genomes versus the number of protein families. And you can plot a rate of recovery of new protein families per genome. Does that make sense, basically? So you're just comparing the genomes to each other. You ask, when I add a second genome, is it all the same protein families, or is it new protein families? Third genome, is it all the same protein families, or is it new? And you get a rate of recovery. And you can do this for different genome data sets. So you can do it for genomes that are closely related to each other, or genomes that are distantly related to each other. So if you do this for Streptococcus agalactiae, This is the species that people discovered the pangenome that we talked about before in. Each genome that you analyze, as you add new genomes, even within a species, you see 121 new families per genome when you do this across. I mean, that's kind of amazing. People didn't really expect this. As you sample more and more genomes within a species, it's like that E. coli story, you see new families. If you do this within a family of bacteria, so as you sample different genomes from across the diversity of the Enterobacteriaceae, the rate of recovery of protein families per genome is higher than the rate when you do it within a species. If you do it within a phylum, the rate of recovery of new protein families is higher. And if you do it across all the genomes we were sequencing, it's the highest. So the further apart organisms are on the ribosomal RNA tree, the higher the probability that they will have different protein families. That's not consistent with this model that lateral gene transfer, that organisms could just sample from all known protein family diversity. It basically means that just like in plants and animals, lots of stuff happens on branches in the evolutionary tree of bacteria and archaea. And inventions happen here that stay in the descendants of that branch. In evolution lingo, these are called synapomorphies, shared derived traits, milk in mammals and so on. And those happen in bacteria and archaea just like they happen in plants and animals. They're not at as high a rate. There is lateral gene transfer, but you have these synapomorphies. Yeah?
The argument that lateral gene, that you wouldn't find new protein families because of lateral gene transfer, I don't understand how that can be postulated, considering that since bacteria tend to have small genomes, how could you not find new protein families?
It's not saying that you wouldn't find new protein families. It's saying that the protein families that you find in an organism are not related to its history, but are related to where it lives. And what this says is that the history matters. The vertical evolutionary history of bacteria and archaea influences the probability that they will have something new. That's all, right? Does that make sense? So anyway, that's the main sort of point here. This has led to all these sort of spin-off projects now. There's a cyanobacterial project. We did a haloarchaea one. And then the last thing, which is to lead into next classes, is, okay, so this is cool. We've sampled 300 genomes across the tree of life. It seemed like we were making this great progress. And then we did this analysis, which is really depressing. So if you zoom in here, what we did here is basically take the tree of life that we use, the ribosomal RNA tree, and then we calculated this PD, phylogenetic diversity, the total branch length for different data sets. So we asked, what is the total branch length covered by the available genomes, the publicly available genomes, before we started this project? And we sorted them by the longest branches. But if you sum all them up, there were like 700 genomes, and the total branch length came to these 20 units, the sort of arbitrary units of total branch length. Then we add our genomes, each of these genome encyclopedia genomes. Each genome we added a lot of phylogenetic diversity. It better have, that's how we selected them, right? So we picked them based upon their novelty. So we added another, 4 or 5 units of phylogenetic diversity. And then if we go back to the tree, and we ask, so here's our new genomes. And if we go back to the tree and we ask, what hasn't been sampled yet? How far have we gotten in sampling the phylogenetic diversity in the bacterial tree? And we first asked for organisms that you could culture. What is left to do? And that's what's shown over here in dark gray. So it's not that bad. We would need to get to about 200 total units of phylogenetic diversity to cover the 10,000. There are only 10,000 described cultured species of bacteria and archaea. We'd only need about 5,000 more genomes to capture half of this. That's doable in genome sequencing land now. It's not totally unreasonable. And that they're doing. So they're now spun off another project to try and sequence all of these. And then, here's the sad part. Then we looked at the branches that came from ribosomal RNA sequencing from environments, the uncultured organisms. And we would need about 20,000 genomes to capture half of the diversity that was known like 10 years ago from the data we got in our database. Right now, it just keeps going up. As I mentioned, every environment that people go to, there's more novel ribosomal RNAs. We've basically sampled with this brilliant genome encyclopedia project like 1/10 of half an eighth of a tenth of a percent of nothing. Really. And it's great, but the scale of the diversity here is just nauseating. And so there's now yet another spin-off project, which is to go through the real diversity, which is in these uncultured lineages, and try and get genome sequences from those uncultured lineages. And, I took out the slide on how to do this. This person, Tanya Wojcicki at the JGI, has been leading this project, and basically what they're doing is going to an environment where they're known to be many novel lineages, and they take samples from those environments and they sort to get individual cells. So they can't culture things, but you can sort them in a flow sorter to get individual cells. And you place these individual cells into little microtiter dishes. And then you lyse the cells and you do ribosomal RNA PCR from those samples. You sequence them and you figure out which little well contains a phylogenetically novel cell. And then you go back and you run what's called MDA, which is whole genome amplification, and you sequence the genome. Now filling in the real phylogenetic diversity. It's still... So she just published a paper on this a few months ago, where they did 300 or so genomes sampled across real phylogenetic diversity. I don't think I have the figure, but we're still at, an eighth of a tenth of a half of a percent, but we're getting there, yeah.
She's speaking at the metagenomics.
She is speaking on Friday at this little journal club over in the Genome Center. When is the talk, do you know? 10, at 10 in the Genome. I can send around the announcement about this project. She's completely amazingly brilliant. And what they showed in her paper is basically sort of an extension of what we had shown, but they found just absurd examples of novel diversity that you find by doing this type of sequencing. Many new genetic codes, for example, in organisms that are out there in the environment. All sorts of weird genes that you never expected to find in bacteria and archaea, and weird processes by starting to sample the true diversity of organisms, which is from the environment. And that's what leads us into what we're going to talk about next, which is metagenomics. That's why metagenomics is really useful, because we don't have to grow things in the lab, and we can actually characterize the genetic diversity in an environment without as much bias. And I think I will just, I have no idea what time it is, but. Oh, okay, yeah.
So have people used the stuff from the EVE project in their sort of estimates of the number of species on the planet, or like, I know they usually grossly underestimate.
Not from the EVE project, but from ribosomal RNA data, people have tried to use that. The genome data will be helpful because it'll help people interpret metagenomic data from environmental samples. But what you need, mostly what they should be using is ribosomal RNA data, and most people ignore that too when they make those estimates.
If you had to estimate, like, sort of the
Number of species?
Yeah.
Hundreds of millions of bacterial and I mean, if you do a plot, like I've seen some people do, of whether or not over time we're starting to lose asymptote, we're not. So as people go to more environments, we find more and more ribosomal RNA diversity. And every study that has been done where people have looked at the genome show that ribosomal RNA underestimates the genetic diversity rather than overestimates. So I don't think it's even, I mean, I think it's very clear. There are tens to hundreds of millions at a minimum. Now, we're never going to, I mean, some point people will characterize all of them, but I mean, just imagine there are pockets in the rock crust in Antarctica that have things that have been isolated for 60 million years. There are going to be thousands of species in each of those pockets, right? And there are 200 million such sites on the planet, and so there's a lot. There's a lot of diversity, but that's not that surprising. The planet's big. Things have been around for 3.5 billion years. If you have a model where there's some isolation of organisms, and not all genes don't get swapped around by lateral gene transfer at infinite rates, I think it's simple to come to that conclusion. I mean, if there are 2 million beetle species, they've only been around for 60 million years, right? There got to be hundreds and hundreds of millions.